Question number 21. The ploidy of the following components represent in this order of sequence respectively. A. Perisperm. B. Pen, that is PEN, primary endosperm nucleus. C. Fertilized egg, that is zygote. D. Antipodals. E. Synergid. F. Secondary nucleus. So in the given options, let's start with A. Perisperm. Perisperm is a remnant of nucellus. Actually, nucellus is a diploid tissue which is present inside the ovule, and as a result of fertilization or after fertilization, the nucellus which is left out is called as perisperm. So whatever the ploidy that is there with nucellus, the same type of ploidy there with perisperm also. And as we know that nucellus is diploid in condition, so perisperm will also be in diploid condition. B. PEN, that is primary endosperm nucleus. This is a structure which is formed as a result of triple fusion. Triple fusion means the fusion of second male gamete which is haploid and secondary nucleus which is present inside the embryo sac. This secondary nucleus is diploid in condition. As there is a fusion between haploid male gamete and diploid secondary nucleus, the resultant structure, that is primary endosperm nucleus, will be triploid in its nature. And one more thing, that primary endosperm nucleus will be converted into endosperm as a result of post-fertilization changes. And this endosperm is a nutritive tissue for the developing embryo which is growing inside the seed. That is, embryo and endosperm, both of them are present inside the seed only. Then third one is fertilized egg. Egg is a female gamete which is haploid in condition, but here it is given as fertilized egg. Fertilized means the one which have been fused with the male gamete. So egg cell is haploid and the male gamete is haploid. If two haploid structures are fused, it resulting into a diploid structure. So here haploid egg is fused with the haploid male gamete to result into zygote. This zygote is otherwise called as fertilized egg. So it is diploid in condition. Next, antipodal. Antipodal is a group of three cells, or we can say three cells are present towards the chelazal end of the embryo sac, which are structurally similar to each other, and these antipodals are considered as the smallest cells of embryo sac and vegetative cells of the embryo sac. Why they are so called as vegetative cells means they does not participate in fertilization at all. That's the reason why these are so called as vegetative cells of the embryo sac. And each antipodal cell in that group of three cells will be haploid in condition. Why they are haploid in condition means they are formed as a result of meiotic division of the megaspore mother cell. So antipodals are haploid in condition. Next, E. Synergid. There are two synergids in the embryo sac along with the egg cell. That is, one egg cell and two synergids will form a group of cells which are present towards the micropyle of the embryo sac, and that three group of cells are called as egg apparatus. And these two cells are also formed as a result of meiotic division of the megaspore mother cell. That's the reason why these synergid cells are also in haploid condition. Then F is secondary nucleus. Okay. In an embryo sac, we know that the best example that is there in our syllabus is polygonum type, in which 7 cells and 8 nucleated structure is present. So 7 cells are 3 cells which are present in egg apparatus, which includes 1 egg cell, 2 synergids, and 3 group of cells which are present towards the chelazal end, which form antipodals. That means 3 cells towards the micropyle and 3 cells towards the chelazal end. And one more cell is present in the center, which is called a secondary nucleus. This secondary nucleus is formed as a result of the fusion of two polar nuclei. One is coming from upper side, one is coming from the lower side. So these two polar nuclei are haploid, which are fused to form a diploid secondary nucleus. So this secondary nucleus is diploid in condition. So from the given options, A is 2n, B is 3n, C is 2n, D is N, E is N, and F is 2N. So in the given options, question number 21, the correct answer is 2. Question number 22, the number of chromosomes involved in double fertilization in Haplopappus gracilis is options 1, 20, 2, 6, 3, 10 and 4, 8. Okay, let's come to the question here. Haplopappus gracilis. In this plant, the diploid number of chromosomes are 4 and haploid number will be 2. That means the diploid number means 2 genomes are there and each genome contains 2 chromosomes. So 2n is equals to 4 and n is equals to 2. Okay, here the question is related to double fertilization. Double fertilization is a unique feature present in angiospermic plants where 2 times of fertilization is taking place. So let's see what are those 2 fertilizations. One, that is called as syngamy process. Syngamy is a fusion of egg cell which is haploid with a male gamete which is also haploid in condition. So this one will be resulting in zygote which is diploid. And the second fusion is called as triple fusion, or second fertilization process is called as triple fusion, where the secondary nucleus of embryo sac which is diploid in condition will fuse with the other male gamete which is haploid in condition to form into primary endosperm nucleus which is triploid in condition. So here 2 times of fertilization is taking place, so we call it as a double fertilization process. Now let us incalculate these values into this one. Okay, egg cell which is n, so n is equals to 2 and male gamete it's also n, it is equal to 2 and zygote 2n which is equals to 4. That means during syngamy process the 2 chromosomes in the egg, 2 chromosomes in the male gamete are taking part. So that for syngamy process total 4 chromosomes are taking part in the fusion. Whereas in triple fission, secondary nucleus which is diploid in condition. So the number of chromosomes are 4 here, male gamete haploid, so the number of chromosomes are 2. So that means a primary endosperm nucleus contains 6 chromosomes. So for triple fission, the total number of chromosomes that are participating are 6. So as double fertilization includes syngamy and also triple fission, the total number of chromosomes which are participating in double fertilization in Haplopappus gracilis is 10. So question number 22, the correct answer is 3. Question number 23, total number of paternal and maternal genomes that are involved in double fertilization respectively are options. First option 2 and 3, second option 2 and 1, third option 3 and 2, fourth option 2 and 5. So in the last question itself we had said that double fertilization is the unique feature present in angiosperms, and in the double fertilization process two male gametes will participate and each of the male gamete is haploid in condition. That means 2 genomes of male parent, that is we can say that paternal genomes are participating, and when it is coming to the female side, okay, egg cell which is haploid and secondary nucleus which is diploid in condition. So that means 1 genome of egg cell and 2 genomes of secondary nucleus are participating, such that a total of 3 genomes of the female parent are taking part in the double fertilization. So in the question they are asking about the total number of paternal and maternal genomes that are involved in the double fertilization are: paternal chromosomes are 2 and maternal chromosomes are 3. So question number 23, the correct answer is 1. Question number 24, in the embryo sac of oryza, just before fertilization has 2 male gametes, 1 synergid, 1 secondary nucleus and 3 antipodals. what will be the total number of chromosomes in it? Options 1, 96, 2, 108, 3, 120, 4, 84. Okay, in the question, it is related to oryza plant. In oryza, diploid number of chromosomes is 24 and haploid number of chromosomes is 12. And here the question is related to before fertilization, it has 2 male gametes in it. Okay, here 2 male gametes. We know that each male gamete is haploid in condition, so that 2 genomes are present. Each genome contains 12 chromosomes, so 2 into 12. It is 24 chromosomes are there in the 2 male gametes. Then second one is 1 synergid. This synergid is haploid in condition, so it contains 1 genome. Each genome contains 12 chromosomes, so the number of chromosomes here it is 12. Third one is 1 secondary nucleus. Secondary nucleus is diploid in condition. Why? Because it is formed as a result of fusion of the haploid upper polar nucleus and haploid lower polar nucleus. So 2 genomes are present, and each genome, as we said, that contains 12 chromosomes. So 2 into 12, 24 chromosomes are there in the secondary nucleus. Then 3 antipodals. Antipodals which are present inside the embryo sac are also haploid in condition, but here they are saying that 3 antipodals are there, so that total number of genomes in all the 3 antipodals are 3. So 3 into 12, it is coming to 36 chromosomes. Then these are the clues that have been given in the question, and based on the clues we got 24 chromosomes in 2 male gametes, 12 chromosomes in 1 synergid cell, 24 chromosomes in 1 secondary nucleus, and 36 chromosomes in 3 antipodal cells. If we add all the chromosomes, 24 plus 12 plus 24 plus 36, we get total number of chromosomes in the embryo sac of oryza before fertilization is 96. Question number 24, the correct answer is 1. Question number 25, how many mitotic divisions are required in the formation of male gametes sufficient for the formation of 120 seeds in an angiosperm? Options 1, 240, 2, 120, 3, 225 and 4, 180. Okay, here 120 seeds. Actually, if you come to the character of seeds, seeds are produced by spermatophyta group of plants. Spermatophyta means, sperma means a seed, phyta means plants. That is, the plants which are producing seeds are called as spermatophytes. All flowering plants are spermatophytes. So flowering plants are gymnosperms and angiosperms. But there is a difference between the type of seeds which are formed, that is naked seeds are present in gymnosperms and the seeds are covered by the fruit in angiosperms. That difference is there between the characters of gymnosperms and angiosperms. Let's come to the number of male gametes that are required for this one and mitotic divisions which are required to produce those male gametes. Okay, in the question you have to produce 120 seeds. When here it is a question related to seeds, we have to consider the male and the female gametes. Male gametes, that is 2 male gametes which are formed in the male gametophyte, whereas egg cell and secondary nucleus which are formed in the female gametophyte or embryo sac. Here the question is about the mitotic divisions which are required in the formation of male gametes only, they are not specifying about the female gametes divisions. Okay, let's see here. To produce 120 seeds, 120 eggs are required, and these 120 eggs are fertilized by 120 male gametes. But one thing to be noted here, that when you are saying about the seed, apart from the zygote, that is embryo, we should also consider the formation of endosperm. That is 120 secondary nuclei will participate in fusion with the 120 other male gametes. That means in total we require 120 male gametes for triple fusion process. So total number of male gametes which are required to produce 120 seeds is 240 male gametes. And there is one relation here. Pollen mother cell which is diploid in condition will undergo one meiosis to produce a tetrad of microspores. That is 4 microspores are present which are haploid in condition. These microspores will form into 4 pollen grains, and each pollen grain will undergo 2 mitosis to form into 2 male gametes. Based on that one, we can say that a total of 8 mitosis will take place to produce 8 male gametes from 4 pollen grains. That means one pollen mother cell is giving rise to 8 male gametes by 8 mitotic divisions. So there is a small relation that always the number of mitotic divisions and the number of male gametes formed will be equal. Okay. So here in the question it is about formation of 240 male gametes. As I said that the number of mitotic divisions and the male gametes are equal, or 1:1 in ratio. So if 240 male gametes are required, how many mitotic divisions are required means it is same the number as like that of male gametes. That means they require 240 mitotic divisions. Okay, from the given options, question number 25, the correct answer is 1, that is 240 mitotic divisions. Question number 26, identify the ratio of male and female nuclei shown in the following diagram that are participating in double fertilization. Options 1, 1:1, 2, 1:2, 3, 2:1, 4, 2:3. In this diagram, the pollen tube is releasing the 2 male gametes, okay, and this is a egg cell and this is a central cell or the secondary nucleus. Okay, here the 2 male gametes, each of them is haploid, so that the number of genomes are 2. Whereas in the female structures, egg cell which is haploid and secondary nucleus or the central cell which contains 2 nuclei, that is upper polar nuclei and the lower polar nuclei. So the number of nuclei which are belonging to the female structures are 3. So that is 3 genomes or 3 nuclei. That means 2 and 3, or we can say the ratio, if they are asking the ratio between male and female nuclei which are participating in the double fertilization that are shown in the figure are 2:3. Question number 26, correct answer is 4. Question number 27, study the following list. List 1, A. Endothecium, B. Tapetum, C. Stomium, D. Sporogenous tissue. List 2, 1. Present between the pollen sacs, 2. Fibrous thickenings, 3. Outer layer of microsporangium, 4. Nourishes the pollen grains, 5. Center of microsporangium. So from that options that have been given, we have to identify the correct match of list 1 and the list 2. Okay, let's come to list 1. A is endothecium. Here whatever the options that have been given, those are related to the anther wall and the anther structure. Endothecium is a second layer present in the anther wall and it is considered to be the hypodermal layer. Hypodermal means which is present just below the epidermis tissue. And this hypodermal endothelial layers or the cells which are present in endothecium contains the specialized radially elongated fibrous thickenings on their walls. So endothecium, the correct option is 2. B is tapetum. Tapetum is the innermost layer of anther wall which is considered to be containing many cells, and each cell is having thin cell wall and abundant cytoplasm inside one with an clearly visible nucleus. And one important thing regarding this endothecium, regarding this tapetum is it is a nutritive tissue. So for whom it is providing the nourishment means it is providing the nourishment to the developing pollen grains which are formed inside the pollen sac or developing from sporogenous tissue. So from the given options we can say that nourishes the pollen grains is exactly suiting to this tapetum. So that is option number 4. C. Stomium. Here in the anther wall, outermost layer is epidermis, and epidermal cells which are present between two pollen sacs which are large in size and contains thin cell wall inside them. Those thin walled large size cells are called as stomium cells, which will help in dehiscence of anther wall when the anther become matured. So that from the given options we can say that stomium is related to 1, that is present between the pollen sacs. Then D is sporogenous tissue. Sporogenous tissue is nothing but the parent cells of microspore mother cells, the parent cells of microspore mother cells, and these sporogenous tissue are present inside the pollen sac, or present towards inner side of the nutritive tissue, that is tapetum. And from the given options we can say that it is a one which is occupying the center of the microsporangium. So that is 5. So that A is 2, B is 4, C is 1 and D is 5. So question number 27, the correct answer is 2. Question number 28, study the following list. List 1, A. Entomophily, B. Ophiophily, C. Caropterophily, D. Therophily. List 2, 1. Snakes, 2. Insects, 3. Squirrels, 4. Birds, 5. Bats. Okay, here in the list 1 and list 2, there is a relation between the type of agent which is helping in pollination. So here in the list 1, first one, entomophily. Entomophily means the pollination with the help of insects is called as entomophily. Of all the biotic agents, the most frequently taking method of pollination is by insects only. So entomophily means by insects, that is 2. B is ophiophily. Ophiophily means the pollination which occurs with the help of snakes is called as ophiophily. So ophiophily is 1. Next, C is caropterophily. Caropterophily means the pollination which occurs with the help of bats is called as caropterophily, that is 5. Then D, therophily. Therophily means the pollination with the help of squirrels is called as therophily. So from the given options we can say that A, entomophily is 2, B, ophiophily is 1, C, caropterophily is 5, D, therophily is 3. Question number 28, the correct answer is 4. Question number 29, study the following list. List 1, A. Porogamy, B. Mesogamy, C. Chalazogamy, D. Emasculation. List 2, 1. Integuments, 2. Stamens, 3. Chalaza, 4. Micropyle, 5. Butter paper. So here in the options, here in the list 1, it is related to, most of them are related to the entry of pollen tube into the ovule, that is porogamy, mesogamy and chalazogamy. Whereas D, emasculation, related to the removal of anthers from the female flower regarding the artificial hybridization process. Now let's come to first one, A. Porogamy. Porogamy means the entry of pollen tube into the ovule through micropyle region is called as porogamy. That means in the given options of list 2, fourth is micropyle. So that suits exactly to the porogamy. The second one is mesogamy. Mesogamy means the entry of pollen tube into the ovule passing through either funiculus, or that is the stalk of the ovule, or through the integuments. So in the list 2, integuments is present. So we can say that it exactly suits to that one, that is B is 1. C is chalazogamy. Chelazogamy means the entry of pollen tube into the ovule through chelaza is called as chelazogamy. This chelazogamy was first identified by a scientist called as Trube scientist, T-R-E-U-B, Trube, in a plant called as Casuarina. So in the list 2, option 3 is exactly suiting to this chelazogamy. Then D, emasculation. During artificial hybridization process, we know that two parents are selected. One is a male parent and other is a female parent. Both the male and female parents are having bisexual flowers. So here, in order to avoid the self-pollination, the stamens or anthers are removed from the female parent. So that process of removal is called as emasculation process. So from the given options, stamens are very much there here, so that emasculation related to stamens. So from the given options, A, porogamy is 4, B, mesogamy is 1, C, chelazogamy is 3, D, emasculation is 2. So question number 29, the correct answer is 3. Question number 30, study the following list. List 1, A, ploidy of binucleated tapetum, B, ploidy of female gamete, C, ploidy of PEN, D, ploidy of microspore and number of genomes in it. List 2, 1, N and 1, 2, N, 3, 2N plus 2N, 4, 3N, 5 is also 3N. In the given options, first one, ploidy of binucleated tapetum. So binucleated tapetum, that is tapetum is an one which contains two nuclei, that is diploid in condition, and it will be having two nuclei. That means 2N plus 2N. Two nuclei are present, each nuclei is diploid in condition. That means A is 3. Second one, B, ploidy of female gamete. Already we had discussed that female gamete is nothing but egg cell which is haploid in condition. So that N. B is 2. Then ploidy of PEN, that is primary endosperm nucleus. So in the given options, there we know that triploid condition is very much there, and the primary endosperm nucleus is formed as a result of triple fusion. So it will be having 3 genomes. So that 3N is the correct one. So 4 and 5, both of them are present. Let's see which one exactly suits to the options that have been given in the question. The D, ploidy of microspore and number of genomes in it. Microspore is nothing but pollen grain which is formed as a result of meiosis. So it will be haploid in condition, and as haploid means N, which contains only one genome. So that N and 1 is correct. That is 1. So given options, in the given options we can say that A is 3, B is 2, C is 4 or 5 and D is 1. So in the given options it exactly suiting to answer number 1. So question number 30, answer is 1. Question number 31, study the following list. List 1, A, microspore, B, tapetum, C, endothecium, D, microspore mother cell. List 2, 1, hypodermis in anther wall, 2, meiocyte, 3, bi or multinucleated cells, 4, first cell of male gametophyte, 5, nutritive tissue of female gametophyte. So in the given options, list 1 and 2, let's see first one, microspore. Microspore is a haploid structure which is formed as a result of meiosis of pollen mother cell. So we can call this as a meiospore. Meiospore. From the options we can say that it's the first cell of the male gametophyte. Why? Because from here onwards the male gametophyte will start its journey and giving rise to two cells, that is vegetative cell and the generative cell, and further the generative cell will divide to form two male gametes. So now after the formation of pollen tube, the 3 cells, that is the vegetative nucleus and 2 male gametes will enter into that one, and that is acting as a male gametophyte. So all the structures are formed from the microspore. So we consider this microspore as a first cell of male gametophyte, that is 4. B is tapetum. Tapetum is the innermost layer of anther wall which is nutritive in tissue. Generally it contains 2 nuclei in each cell, or sometimes more than 2 nuclei also, and it is nutritive in function. So based on these characters, let's see which character is related to the tapetum in the list 2. So in the list 2, third option is bi or multinucleated cells are present. So that exactly suits to tapetum. Then C is endothecium. Endothecium is a second layer of cells present in the anther wall, which is present just below the epidermis. As it is present just below the epidermis, it is acting as a hypodermal tissue, and it is having fibrous thickenings on their wall, and it also help in the dispersal of the spores. So from the list 2, which character is related to endothecium is hypodermal in anther wall, that is 1. And D is microspore mother cell. Microspore mother cell is a mother cell of microspores, which is diploid in condition, and this microspore mother cell, otherwise called as pollen mother cell, will undergo meiotic division, and any cell which is undergoing meiotic division, we call it as meiocyte. So in the list 2, it is very much related to microspore mother cell, that is meiocyte. So if we match the characters of list 1 to that of the characters of list 2, A microspore is 4, B tapetum is 3, C endothecium is 1, and D microspore mother cell is 2. So question number 31, the correct answer is 1. Question number 32, study the following list. List 1, A allium, B solanum, C helianthus, D hibiscus. List 2, 1 protandrous flowers with anatropous ovule, 2 monothecous anthers and hercogamy, 3 protandrous flowers in umbel inflorescence, 4 monocacial cyme with flowers showing protogyny, 5 heterostyle in cleistogamous flowers. So let's come to list 1, that is allium. A allium. Allium is a plant which is exhibiting a protandrous condition, that is in a bisexual flower. If androecium matures earlier than gynoecium, we call it as protandrous condition, and such flowers are considered to be the protandrous flowers and protandry also. And allium is a plant which is forming inflorescence called as umbel inflorescence. Umbel inflorescence in which the peduncle is condensed, and on that condensed peduncle the flowers will develop in centripetal manner, that is from periphery to the center. Periphery to the center. So A allium, that is protandrous flowers with umbel inflorescence. B is solanum. Solanum is a plant which belongs to Solanaceae family, and this shows the cymose type of inflorescence. In the cymose type of inflorescence, the peduncle growth is restricted by formation of an flower at the tip of the peduncle, and the further flower formation will be taking place towards the downside. So that is oldest flower, the first formed flower will be at the top, and the last formed flower will be at the base of the peduncle. And here in solanum, one special feature is there. Every time one branch is developing from the base of the flower, and that branch is also ending with one individual flower. So likewise, every time one branch is formed, so we call it as a monocacial cyme. And one more character related to solanum is the cross pollination controversy exhibited by it is protogyny. Protogyny is a part of dichogamy condition where maturation time of male and female reproductive organs is different, in which protogyny means the maturation of gynoecium earlier than androecium. So that solanum exhibits monocacial cyme with protogynous condition, that is 4. Then C is helianthus. C helianthus, which belongs to Asteraceae family. The common name of helianthus is sunflower. This exhibits a controversy of cross pollination where maturation period of androecium and gynoecium is differing in a bisexual flower. So here it is called as dichogamy condition. In this dichogamy also, these flowers exhibit a case where androecium will mature earlier to gynoecium, so we call it as protogyny nature. Then one more thing is the type of ovule here. So here the ovule of all Asteraceae members will have inverted nature. that is the degree of curvature exhibited by them is 180 degrees. So the one which is exhibiting 180 degree of curvature, we call such a ovule as anatropous ovule. So Helianthus is 1. D. Hibiscus. Hibiscus belongs to Malvaceae family, and all Malvaceae members will exhibit a special feature related to anthers or stamens, that is monothecous nature. Monothecous means the anther is having only one lobe in it, or the stamen contains only one anther lobe in it. So inside one anther lobe, as we know that 2 pollen chambers are present, so we call this as a bisporangiate type of character. So second one is here hibiscus will exhibit a cross pollination controversy where andrecium and gynoecium, or the stamens and stigma, are at different levels. And one more thing here, it is the stigma are protruded beyond the anthers. Because of this nature, there is no chance for self pollination, they have to go for cross pollination only. So from the given options, D is 2. A. Allium is 3, B. Solanum is 4, C. Helianthus is 1, D. Hibiscus is 4. So question number 32, the correct answer is 1. Question number 33, study the following list. List 1, A. Zygote, B. Perisperm, C. Endosperm, D. Nucellus. List 2, 1. Naked megasporangium, 2. Product of triple fission, 3. Product of syngamy, remnant of nucellus, 5. Seed coat. So in the given options, let's come to the conclusion part, that is A. Zygote. Zygote is a first cell of sporophytic generation, that is a first diploid structure in the life cycle, and it is formed as a result of fusion of male gamete and a female gamete. And this is a one which is formed as a result of syngamy, that is what we call it as a product of syngamy, that is 3. And B is perisperm. Perisperm is nothing but the remnant of a nucellus, that is after fertilization process, the nucellus will be acting as perisperm. So the ploidy of this perisperm is 2n as like that of nucellus. So we can say it's a remnant of a nucellus, that is 4. C is endosperm. Actually to say, endosperm is a structure which is formed from primary endosperm nucleus. That means primary endosperm nucleus is converted into endosperm as a result of post-fertilization changes. And how does this primary endosperm nucleus is formed? It is formed as a result of fusion between haploid male gamete and a diploid secondary nucleus, and that process is called as triple fusion. So we can say that it is a product of triple fusion, that is 2. And D is nucellus. Nucellus is nothing but the diploid tissue which is present inside the ovule, and this nucellus contains embryo sac inside it, and the nucellus is surrounded by a protective sheet called as or protective layers, we can consider those are called as integuments. And if that integuments are not present, we call it as a naked megasporangium. So that nucellus is 1. So from the given options, A. Zygote is 3, B. Perisperm is 4, C. Endosperm is 2, D. Nucellus is 1. So question number 33, correct answer is 1. Question number 34, assertion. Pollen grains are well preserved as fossils. Reason. The sporopollen of exine is one of the most resistant organic material known and can withstand high temperature, strong acids and alkali. Okay. As we know that pollen grain is the first cell of male gametophyte which is haploid in its nature. Pollen grain contains 2 layers, exine, outer one, and inner one, intine. Exine is made up of a chemical called as sporopollenin, whereas intine is made up of a chemical called as pectocellulose. Outer wall, that is exine, which is thick, and here and there they contains a part where circular regions we can call them as, where the sporopollen is absent, those are called as germ pores. And the pollen grains are well preserved in the form of fossils. That means they can retain for years together in the form of fossils. What's the reason for that retaining as a fossil means? Because of the sporopollenin which is present in the exine wall. So what is the specialty of this sporopollenin means? It is having resistant to high temperatures, it can resist for strong acids and alkali also. And one more thing, that it can resist for any type of enzymatic activity. That means any enzyme can't do anything to this pollen grain due to presence of sporopollenin. So in the given options, assertion A is correct, reason R is also correct, and reason is giving the correct explanation for assertion. So question number 34, correct answer is 1. Question number 35, assertion both autogamy and allogamy occur in Viola, Oxalis and Commelina. Reason, they contain flowers which open and similar to flowers of other species with exposed anthers and stigma, and the flowers which do not open at all. So that means it contains both the type of flowers in the options. Okay, let's come to the 3 plants, that is Viola, Oxalis and Commelina. Here in these 3 plants, 2 types of flowers are present in each plant. Those are called as chasmogamous flowers and cleistogamous flowers. Chasmogamous flowers means the flowers which will open are called as chasmogamous flowers, whereas the cleistogamous flowers is the flowers which never open are called as cleistogamous flowers. Okay, here chasmogamous flowers, as like that of other plants, here the essential organs are very much exposed to the environment and they will carry on the cross pollination as like that of other plants. But when it is coming to cleistogamous flowers, the essential organs are enclosed inside the perianth parts. They are not exposed to the outer environment, so there is no chance of cross pollination. They have to go only for self pollination process. The self pollination which is taking place in the cleistogamous flowers is called as cleistogamy, whereas the pollination which is taking place in chasmogamous flowers is called as chasmogamy. So both autogamy and allogamy is possible in Viola, Oxalis and Commelina. Why? Because in chasmogamous flowers it is cross pollination, that is allogamy. In cleistogamous flowers it is self pollination, that is autogamy. Both of them are possible here. Why? Because in chasmogamous flowers the essential organs are exposed, whereas in cleistogamous flowers the essential organs are enclosed. So because of this one we can say that both are possible in the 3 plants. So question number 35, the correct answer is 1. Question number 36, assertion coconut water is a free nuclear endosperm and the surrounding white kernel is a cellular endosperm. Reason, primary endosperm nucleus, that is PEN, undergoes free nuclear division followed by cell wall formation. So here if you come to the coconut, in coconut we know that 2 types of endosperms are present. One is called as a liquid endosperm and other is called as a solid endosperm. So let's come to first liquid endosperm. So liquid endosperm means the one which contain only nuclei in it, no cells. That means how does this liquid endosperm is formed? It is formed as a result of free nuclear divisions without the division of the cytoplasm. And there is one more endosperm is there, that is called as a solid endosperm. That is what we call it as a kernel, called as kernel. So the one which we eat. And how does this solid endosperm is formed is, after some time, when we say that first free nuclear division is taking place, after some time each nucleus will start accumulating some cell wall around it and cellularization will be taking place, or we can say cell wall formation will be taking place. So this is because of the cellularization process or cell wall formation. So that the liquid endosperm is resulted because of free nuclear division and solid endosperm is resulted because of cell wall formation or cellularization process of the primary endosperm nucleus. So question number 36, the correct answer is 1. Question number 37, assertion a dithecous anther becomes 2-chambered towards maturity. Reason, sterile partition between the adjacent theca disintegrates in a matured anther. Let's come to that first point. So here in ditheca anthers will become two chambers at maturity, means we know that thin-walled large size cells are present between the pollen chambers called as stomium cells. Those stomium cells, when they disintegrate, so the pollen grains are liberated, but before that process, whatever the wall layers which are present between the pollen sacs will be disintegrated. Because of that one, the two chambered cavity will be converted into one chamber, so that in total 4 pollen chambers are converted into two chambered structure. That is the dithecous anthers become two chambered or bisporangiate towards the maturity. So that is correct. But this in the second option, that is a reason, it is given a sterile tissue between the adjacent theca is actually it is not that tissue, but it is a tissue which is present between the pollen chambers is disintegrated. So reason is incorrect. So that A is true and R is false. Question number 37, the correct answer is 3. Question number 38, assertion. Chromosomal number is reduced to half during microsporogenesis and megasporogenesis. Reason. Chromosomal number is reduced to half by equational division. Okay, here in the assertion statement, it is related to microsporogenesis and the megasporogenesis. Let's come to the microsporogenesis. What do you mean by microsporogenesis? Means the formation of microspores is called as microsporogenesis. When does this microsporogenesis taking place? Means here the microspore mother cell or pollen mother cell will undergo meiosis. It forms a tetrad of microspores. That means the diploid pollen mother cell will undergo meiosis to form into tetrad of microspores. This process is called as microsporogenesis. That means here the type of division is reduction division, not equational division. And when it comes to the megasporogenesis, here also the megaspore mother cell, which is diploid in condition, will undergo meiosis to form into tetrad of megaspores. So the diploid megaspore mother cell will undergo meiosis to form into haploid tetrad of megaspores, and this process is called as megasporogenesis. And here also what is happening is a meiotic division is taking place, or we can call it as a reduction division. And as and when the reduction division or meiosis is taking place, we know that chromosome number is reduced to half. So 2n is converted into n. Here also 2n is converted into n. So that during microsporogenesis and megasporogenesis, the chromosome number is reduced to half. So assertion statement is correct. But when it is coming to the reason statement, it is given that chromosomal number is reduced because of the equational division. Equational division means mitosis. In mitosis, never there will be a change in the number of chromosomes. Whatever the number of chromosomes that are there in the parent cell, the same number of chromosomes are present in the two daughter cells formed from that parental cell. When does there is a change in the number of chromosomes? Means when meiotic division or reduction division is taking place. And in the meiosis, during which phase does the chromosome number is reduced to half? Means during anaphase 1, the chromosome number is reduced to half. Why? Because there is a segregation of the genomes, or we can call it as a disjunction process. Because of the disjunction or segregation of genomes, the chromosomal number is reduced to half. So based on the two statements, we can say that assertion statement is correct and reason statement is false. Question number 38, the correct answer is 3. Question number 39, assertion the fertility of pollen grains formed from their mother cell is 100%. Reason, all the 4 pollen grains formed from each mother cell develop into 4 male gametophytes. Let's come to assertion statement. So here we know that pollen mother cell will undergo meiosis to form 4 microspores, and each of the microspore, that is pollen grain, will give rise to 2 male gametes, and that is nothing but the male gametophyte. That means all the 4 which are formed from one pollen mother cell are fertile. That means we can say that it is 100% fertility is present. So the assertion statement is correct. Then coming to the reason. So already we explained why it is 100% fertile, means all the 4 pollen grains formed from the pollen mother cell are fertile in their nature, giving rise to male gametophytes. That means one pollen mother cell will give rise to 4 microspores or pollen grains, and each of the pollen grain will give rise to one male gametophyte. That means in total 4 male gametophytes are formed. Such that we can say question number 39, both A and R are correct and R explains A. Question number 39, correct answer is 1. Question number 40, assertion, in Vallisneria, autogamy and geitonogamy are not possible. Reason, in Vallisneria, both the sporophyte and gametophyte are dioecious. Vallisneria. Vallisneria is an submerged rooted hydrophyte which is dioecious in its nature. Dioecious means here male flowers are present on one plant, female flowers are present on another plant. It is dioecious. And dioecious nature, when you are saying male flowers on one plant and female flowers on one plant, it is related to the sporophyte generation. Sporophyte is dioecious, and in this plant the gametophyte, as we know that all angiosperms are heterosporic in their nature, that is microspores are there and megaspores are there. Microspore will give rise to male gametophyte and megaspore will give rise to female gametophyte. So that gametophyte is also dioecious in its nature. Okay, when dioecious condition is there in a sporophyte, there is no possibility of autogamy, because the primary condition for autogamy is presence of a bisexual flower. So this in Vallisneria, flowers are unisexual, so there is no chance for autogamy. So that autogamy is not possible in Vallisneria plant. Next one is geitonogamy. Geitonogamy is a condition where the pollination which is taking place between the flowers which are present on the same plant. So that here the pollination should occur between the flowers of the same plant, means at least the flower should have 2 types of unisexual flowers on the same plant, otherwise 2 bisexual flowers should be there. But in Vallisneria, one plant contains only male flowers, one other plant contains only female flowers, so there is no chance for geitonogamy also. So assertion statement is correct. And the second one, already we said that in the reason statement is given that sporophyte is dioecious and the gametophyte is dioecious. Because of the dioecious nature, there is no chance for geitonogamy condition. So question number 40, the both assertion reason are correct and reason explains assertion. Question number 40, correct answer is 1. Okay. Question number 41, assertion, in yucca, cross pollination is brought about by moth Tageticula eucacella. Reason, in the absence of this moth, self-pollination fails. Here a plant called as yucca. Yucca belongs to Liliaceae family, which is a monocot, and here in this plant we can say symbiotic type of pollination is taking place. That means it is the agent or the moth which is responsible for this pollination in this plant is very much specific. That means if that moth is not available, it does not participate in cross pollination, but it will participate in self pollination process. That means here the moth which is taking part in the pollination is Tageticula eucacella. So the correct statement related to assertion. But when it is coming to reason statement, so in absence of that moth, cross pollination does not take place, but it will go for the next chance or next option, that is a self pollination process. But in the reason it is given that in absence of this moth, self pollination fails. So that statement is incorrect. Question number 41, the correct answer is 3. Question number 42, assertion, in black pepper and beet, the seed is occupied by perisperm. Reason, seeds of black pepper and beet show persistent remnants of nucellus. Okay, here in black pepper and beet, there is one special feature that the nucellus is left out in a ripened seed. That left out and remnant nucellus is called as perisperm, which occupy majority of the seed. So that assertion statement is correct. And why it is so means, as already I said that the perisperm is nothing but the persistent one which is surviving after the ripening of the seed also, and it's a remnant of the nucellus. So both A and R are correct, and R explains A. Question number 42, the correct answer is 1. Question number 43, assertion, in angiosperms water is essential for syngamy. Reason, in angiosperms 5 genomes are involved in double fertilization. Okay, here angiosperms are highly evolved plants on the earth which are highly adapted for terrestrial habitat, and here this is one of the reason why these are adapted to the terrestrial habitat is there is no need of water for fertilization process. Okay, this character is seen only up to pteridophytes where fertilization requires water, because the male gametes are motile, they will swim in the water and reach the female organs. But from gymnosperms onwards and in angiosperms also, the male gametes are non-motile. That means they are not zooidogamous in the nature, but these male gametes are carried by the pollen tube. That's the reason why there is no need of water for the syngamy process. So assertion statement, it is given that water is essential for syngamy. That statement is incorrect. Whereas the second one, in angiosperms 5 genomes are involved in double fertilization. Right. That is 1 XL haploid fused with 1 male gamete which is haploid. So 2 genomes are involved in syngamy. Whereas during triple fission, 1 male gamete which is haploid and 1 secondary nucleus which is diploid. So 3 genomes are involved. In total 5 genomes are involved in the double fertilization process. So question number 43, the correct answer is 4.